Good afternoon. My name is Joseph Marino. This is Bio 525, Concepts of Medical Microbiology. My topic is Mycobacterium tuberculosis and understanding the pathogen, its impact, and control measures. Introduction. Overview of Mycobacterium tuberculosis. The introduction to M. tuberculosis. It is the bacterium responsible for tuberculosis, otherwise known as TB, a serious infectious disease primarily affecting the lungs, but capable of impacting other parts of the body such as kidneys, spine, and brain. TB is one of the oldest known diseases and remains a significant global health challenge. Significance in public health. TB is a major public health concern due to its high morbidity and mortality rates. It is one of the leading causes of death from infectious diseases worldwide. The disease is spread through the air when people with active TB in their lungs cough, sneeze, or speak, making it highly contagious. Despite being preventable and curable, TB continues to pose a threat, especially in low and middle income countries where healthcare resources may be limited. The purpose of this presentation is to provide a comprehensive overview of M. tuberculosis, its impact on public health, and the ongoing efforts to control and eliminate TB. We will explore bacterium biology, the disease's pathogenesis, current diagnostic methods, treatment options, and the challenges faced in TB control. By understanding these aspects, we can better appreciate the importance of continued research, public health initiatives, and global cooperation in the fight against tuberculosis. Nomenclature and discovery. The common name for Mycobacterium is the tuberculosis bacterium, which is the pathogen responsible for causing tuberculosis, a serious infectious disease that primarily affects the lungs, as I mentioned earlier. This name reflects its classification within the genus Mycobacterium, which includes other pathogenic species such as Mycobacterium bovis and Mycobacterium leprae. It's been discovered by Robert Koch in 1882. M. tuberculosis was discovered by the German physician, and he was a German physician and microbiologist, which was discovered on March 24, 1882. Koch's identification of the bacterium was a groundbreaking achievement in medical science, as it provided the first clear evidence that a specific microorganism can cause a specific disease. The historical context, the importance of Koch's discovery in the field of microbiology. He proved germ theory. Koch's discovery was pivotal in providing the germ theory of disease, which posits that microorganisms are the cause of many diseases. This theory was a significant departure from the previously dominant miasma theory, which attributed diseases to bad air or miasmas. Koch's postulates. Koch developed a set of criteria known as Koch postulates to establish a causative relationship between a microbe and a disease. These postulates became fundamental principles of microbiology and are still used today to identify the causative agents of various infectious diseases. Now, the advancements in laboratory techniques. Koch introduced several innovative laboratory techniques, including the use of solid media to culture bacteria, which allowed for the isolation and identification of specific pathogens. His method laid the groundwork for modern bacteriology. Its impact on public health. Koch's work had a profound impact on all public health practices. By identifying the causative agents like diseases like tuberculosis, cholera, and anthrax, he enabled the development of targeted strategies for disease prevention and control. A Nobel Prize. In recognition of his contributions to medical science, Robert Koch was awarded the Nobel Prize in Physiology or Medicine in 1905 for his research on tuberculosis. Koch's discovery of Mycobacterium has not only revolutionized the field of microbiology, but also has had lasting implications for the diagnosis, treatment, and prevention of infectious diseases. Taxonomy, the hierarchy of biological classification, which is responsible. This presentation will also help outline the taxonomic classification of Mycobacterium. Okay, it belongs to the domain bacteria with the phylum Actinomycetota. The class is Actinomycetia, and the order of Mycobacterialis and family Mycobacteriaceae. The genus is Mycobacterium, and the specific species is M. tuberculosis. Phylogeny of the genus Mycobacterium. Okay, there's many doubts, few certainties. Okay, Enrico Totoli explores the complex and often uncertain evolutionary relationships within the genus Mycobacteria. My research and study also highlights several key points. The complex phylogeny, the genus Mycobacterium includes both slow-growing and rapid-growing species, which are phylogenetically distinct. However, the evolutionary relationships among these species are not fully resolved, leading to many uncertainties. Then you have genetic analysis. The research utilizes genetic data, particularly from 116S RNA, RNA sequences to construct phylogenetic trees. These trees help in understanding the genetic relationships and evolutionary history of different Mycobacterium species. You have taxonomic challenges. Through my research and studying, okay, I will also discuss the difficulties in classifying Mycobacterium species due to their genetic diversity and the presence of many intermediate forms. This makes it challenging to draw clear boundaries between species. Clinical relevance. Understanding the phylogeny of tuberculosis is crucial for clinical microbiology, as it can impact the diagnosis and treatment of diseases caused by these bacteria, such as tuberculosis and leprosy. Overall, the article underscores the need for further research to clarify the phylogenetic relationships within this important genus. Then you have the structure of Mycobacterium tuberculosis, the cell wall, the shape, the staining, the growth characteristics. The cell wall of Mycobacterium is rich in mycolic acid, which gives it a distinct waxy coating. This waxy layer is crucial for the bacterium's resistance to desiccation and many antibiotics. The shape. M. tuberculosis is rod-shaped, also known as bacillus. This shape is typically for many bacteria aids and its identification under a microscope. Then you have the staining. Due to its waxy cell, M. tuberculosis is acid-fast. This means it retains certain strains even after being washed with acid solutions. The Ziehl-Neelsen stain is commonly used to identify acid-fast bacteria, making a key diagnostic tool for tuberculosis. Growth characteristics. Tuberculosis is aerobic, meaning it requires oxygen to grow. It is also slow growing, which can make it challenging to culture in a laboratory setting. Additionally, it's non-motile, meaning it does not have the ability to move on its own. These characteristics are essential for understanding the biology and pathology of M. tuberculosis, as well as developing effective diagnostic and treatment strategies. Pathogenesis, infection mechanism, virulence factors. You have the inhalation of airborne droplets. Tuberculosis, TB, infection begins when a person inhales droplets containing the bacterium M. tuberculosis. Phagocytosis by alveolar macrophages. Once in the lungs, the bacteria are engulfed by alveolar macrophages, a type of immune cell. Evasion of destruction. Despite being engulfed, M. tuberculosis can evade destruction within the macrophages. Formation of granulomas. The immune system responds by forming granulomas, which are clusters of immune cells that attempt to contain the infection within the lungs. Virulence factors. Mycolic acid, cord factor glycolipids. These components of bacterial cell wall contribute to the bacterium's ability to cause disease. You have the inhibition of phagosome and lysosome fusion. And tuberculosis can prevent the fusion of phagosomes, which are vesicles containing the bacteria, which lysosomes are the vesicles containing digestive enzymes, allowing it to survive inside macrophages. Then you have the secretion of proteins. The bacterium secretes proteins that modulate the host immune response, helping it to persist and cause disease. These mechanisms and factors highlight how M. tuberculosis is able to infect and survive within the human body, making TB a challenging disease to treat and control. Epidemiology. You have the prevalence, transmission, and global impact. First, you have prevalence. Tuberculosis has a high incidence rate, especially in developing countries. It's the leading cause of death from a single infectious agent. Transmission. TB spreads through the air when an infected person coughs, sneezes, speaks, or sings. Close contact with an infected individual increases the risk of transmission. Global impact. According to the World Health Organization, identifies TB as a significant global health issue with a particularly severe impact in low-income regions. You can see tuberculosis global epidemiology. You have 8 million new cases per year. You have 2 billion infected, 20 million cases at all times, and 2 million deaths per year. You have the treatment. First line drugs, second line drugs, challenges, and new treatments. First line drugs, isoniazid, a key antibiotic used in the treatment of TB, effective in killing actively growing TB bacteria. Rifamycin, another primary antibiotic, that works by inhibiting the synthesis of bacterial RNA crucial for TB treatment. Ethambutol, used to prevent the development of drug resistance. It works by inhibiting the synthesis of the bacterial cell wall. Pyrazinamide, effective in killing dormant TB bacteria. It helps to shorten the duration of TB treatment. Then you have second line drugs, fluoroquinolone, a class of antibiotics used when first line drugs are ineffective. They work by inhibiting bacterial DNA synthesis. Injectable agents, for example, amikacin. These are used for more severe cases of TB, particularly when resistance to the first line drug is present. Amikacin works by inhibiting protein synthesis in bacteria. Then you have challenges, to multi-drug resistance, MDR strains. These are TB strains resistant at least to isoniazid and rifamycin, and which are the two most potent TB drugs. Extensively, drug-resistant strains. These strains are resistant to first-line drugs and at least one second-line injectable agent and one fluoroquinolone, making them much harder to treat. You have new treatments as well. Bedaquinolone, a newer drug that targets the ATP synthase enzyme in TB bacteria, crucial for their energy production. Then you have delaminid, another recent addition to TB treatment. It works by inhibiting the synthesis of mycolic acids, essential components of the bacterial cell wall. These advancements and challenges highlight the ongoing efforts and complexities in effectively treating TB, especially with the emergence of drug-resistant strains. Here you have medications, you have the different strains, you have the effective drugs, and then you have the resistant bacteria. You have prevention, signs and symptoms of tuberculosis, vaccination, public health measures. Vaccination, the Bacillus calmette-guerin (BCG) vaccine is used to prevent tuberculosis. It is particularly effective in protecting young children against severe forms of TB, such as TB meningitis and malaria TB. Public health measures, early detection and treatment. Identifying TB cases early and providing prompt treatment is crucial to prevent the spread of the disease. This involves screening high-risk populations and ensuring access to diagnostic services. Isolation of infectious patients. Patients with active TB, especially those with pulmonary TB, should be isolated to prevent transmission to others. This is particularly important in the healthcare settings and during the initial phase of treatment, when patients are most infectious. Improve ventilation in crowded settings. Enhancing ventilation in places where people gather, such as hospitals, schools, and public transport, helps to reduce the concentration of airborne TB bacteria, thereby lowering the risk of transmission. These measures are essential components of TB control strategies, aiming to reduce the incidence and spread of tuberculosis in the community. I will now conclude my presentation with an overview of Mycobacterium tuberculosis, the global significance of tuberculosis, objectives of the presentation, the historical importance of Robert Koch's identifying tuberculosis, and a comprehensive exploration. My presentation provided a comprehensive overview of Mycobacterium tuberculosis and its implications for public health. I delved into the global significance of TB as a health concern, its modes of transmission, and the presentation's objectives, which encompassed an exploration of the bacterium's biology, disease pathogenesis, diagnostic techniques, treatment options, and challenges in TB management. Furthermore, it underscored the historical importance of Robert Koch's identification of M. tuberculosis and its profound impact on the fields of microbiology and public health protocols. Thank you for your undivided time and attention. God bless you and have a wonderful evening.